Well, welcome everyone to our Dunleavy June lecture series, and just a few updates that we usually have before we launch in with our guest speaker. I'd like to congratulate our Spring 2026 Seed Grant awardee, Dr. Danielle Pinelli, for development and validation of a digital coaching app to scale evidence-based walking therapy for perinatal depression and anxiety. It's known as the PACES study. So congratulations, Dr. Pinelli. Just to save the date, the fall 2026 Dunlevie MFM Center Seed Grant Cycle will open for applications on Monday, August 17th. We will extend up to 2 awards, $35,000 each, to support research, with the time there from January 2027 to December 31st, 2027. And details are available at the website. So please check it out when you have a chance. We won't be having meetings in July and August, so our next center meeting will be on September the 10th, and we'll be hosting Dr. Anka Pasca, who is an assistant professor in the Department of Pediatrics here at Stanford, for identification of neuroprotective compounds for hypoxic brain injury using high-throughput screening platforms in cortical organoids. Dr. Pasca has been awarded Dunleavy Seed Grants a couple of times for her work, and so we're very excited to hear this lecture and see the progress that she is making in her research. Today, our honored speaker is also our Stanford very own Dr. Kristy Redhorse, professor in the Department of Biology at Stanford University, and her talk is studying the placenta and modeling pregnancy complications in 3 dimensions. And I will hand things over to my colleague, Dr. Virginia Nguyen, for a formal introduction.
All right, welcome everyone, and hope everybody is having a great day. It is my distinct pleasure to introduce Professor Redhorse, who is in the Department of Biology as well as the Stem Cell Institute, and she is a Howard Hughes medical investigator. I have known Kristy since she was a graduate student. So her work started in actually chemokine and chemokine ligand biology in the human placenta. As her postdoctoral work, she was at Genentech, followed by Stanford, becoming a card-carrying vascular biologist, and has been credited with determining the developmental origins of the cells that develop into the coronary arteries, balking some of the dogma of the field, which has given her great notoriety. She is doing phenomenal work in heart vasculature biology and understanding collateral vessel formation with an eye to improve heart health, particularly after an MI. But I'm super excited because Kristy has returned to the placenta, bringing her vascular biology expertise and getting to collaborate with her again after all these decades. So with that, I will turn it over to Kristy to tell us about placental vasculature.
Great. Can you hear me and see my slides?
It looks perfect, Kristy.
Great. Thank you so much for this opportunity to talk to this community. I'm really excited about getting back into the placenta. So the last time I talked to this group, I talked about my heart work. But as Virginia mentioned, I actually have a background in reproductive biology. When we were in the lab together in Susan Fisher's lab, I got a master's and a PhD there, and I studied things like mapping which cells at the maternal fetal interface are expressing chemokines and their receptors. These are molecules that cells secrete and use for cell-cell communication to organize migration and morphogenesis. And I also was able to interrogate some cell adhesion and cell repulsion receptors and do some in vitro modeling of human placental cells to try to gain evidence for the role of these molecules in establishing the tropism of trophoblasts for arteries rather than veins. And excited by this study that I did, then I was triggered to try to do some in vivo modeling of human placental invasion, trophoblast invasion. And so before I left the lab, I completed this study where we put human placental cells into the fat pad and other locations in mice. And we were able to see that trophoblasts invade and target the vasculature anywhere they are, even if it's in another organism. And so that was pretty fun to see. And also, they have this tropism for arteries versus veins. Specifically, they can induce apoptosis in the arteries, but even though they're touching the veins, they wouldn't apoptose or remodel those vessels. And so I had a lot of fun studying this during my PhD. But I really had this desire to have a more tractable system in terms of being intact. And so I wanted to go in vivo and study systems that I could study within the organism, because as you know, the vasculature needs blood flow. And when you model those in vitro, it doesn't quite have all the aspects, especially 25 years ago. And so I changed fields. So for my postdoc and for my own lab, I worked in a different system in the heart because dogma at the time, and also now I've noticed coming to these meetings, that dogma in the field is still that mouse placentation is not a very good model of human placentation. So I went on to study the heart, and in my lab, what we've done over the years is, as Virginia said, mapped what the progenitors are and how they invest the heart as they're making the coronary vasculature. We've done a lot of work to identify how those progenitors will build the arteries, and we've identified a lot of genetic pathways that are regulating this process. We described how these can expand if the heart grows. And then finally, taking these lessons that we learned from development, reestablishing them in the adult so that we can improve recovery and specifically grow these collateral arteries or these natural bypasses. And so I just want to make the point here is that all this work was really bolstered by this vision when I first started my own lab, that looking in 3D is really important. One example is that these collateral connections, you can only see if you trace them in 3D. There's no molecular marker to distinguish these bypasses from the adjacent regular arteries. And so in doing that type of analysis, we were able to identify this chemokine, CXCL12, and this is its receptor, CXCR4, as being really important for coronary artery development. And when you reestablish it in adult after recovery, you can grow these collaterals. And so we have been invested for many years, and most recently have shown that this pathway regulates the development and architecture of collateral arteries in the human heart. And I just want to make a big point here is why I love Stanford. So this has been a very exciting part of my lab in the past few years, and it was triggered by Andrew Goldstone, who saw the way that we were dissecting development and said, you need to take this to a type of vessel that's actually going to be useful in the clinic. And so with Andrew Goldstone and people that came after him in the lab, we focused on taking our methods into these collateral arteries. And so I'm really excited to get to work with this community and hopefully have some similar successes. And we've been working with Virginia in her lab for a bit now, and it's been great. And part of that has been Team Placenta in my lab now. So this is work that I'm going to show you today that is carried out by James Wierzinski in the lab. He's a third-year PhD student, and he's mentored by the Stanford Science Fellow, Mira Mufarej, and myself. And the data that I'm going to show you from the study today is on bioRxiv, and we have a really close relationship, this team, and we talk a lot and plan a lot together, but every experiment you'll see today from me and in this paper was done by James. Okay, so this audience knows that pregnancy complications are a really important thing to focus on in research, and that they arise from, or they're thought to arise from, problems in placentation that then sequelate into maternal cardiovascular problems. And so as we all talk about in this field, and as is kind of the standard thought, it's that trophoblast invasion and remodeling of the arteries at the maternal-fetal interface is either too little or too much. And so we talk about in these meetings frequently how trophoblasts are going to invade the maternal vasculature, and this is inhuman. And when they don't invade enough or cause the remodeling of the vessels, there's too little preeclampsia growth restriction. And then if there's too much, then you have placenta accreta, which, as we heard from Dr. Afshar's talk a few months ago, there is some evolution in the thought of what causes this, and it's likely looking and like it's more due to a decidual defect rather than over-invasion propensity of the placental cells. But nonetheless, this is very scary. We all have heard and talked about how the placenta stays attached at birth, and there's a hemorrhage and many other things that are really scary for the woman. But there's few treatments for pregnancy complications, and there's many reasons for that, but some things that maybe my lab and our particular type of research can help address is the fact that we lack mechanistic understanding of many of the aspects of normal placental development, that there is a lack in the mechanistic understanding of the pathology, which is related to this fact that the pathology is instigated early in pregnancy before you even know that a woman will develop one of these. But then you can only understand and get tissue to do experiments with and analyze for the most part later on. And so that is a real roadblock, we believe, to understanding the mechanisms. Another roadblock is this lack of this animal model, because you need intact systems to really understand some important mechanistic parts of how tissues are working and developing. And so this is just a quote from a mouse placenta researcher and saying that, and this is a sentiment that's said all the time in the literature currently, there's no evidence for bulk invasion of trophoblasts, and there's really not enough parallels between the mouse and human. So today I'm going to tell you how we have most recently in this publication here started addressing some of these issues. And I'll tell you how we took this whole organ 3D imaging and tried to readdress whether mouse is a good model for studying some of these aspects of placental development. I'm going to tell you about this really important pathway that keeps coming up and that how it's also important in spiral artery development. And then I'm going to tell you if this signaling pathway is inhibited then early in pregnancy, then it causes a placenta accreta-like phenotype in mice. So whole organ 3D imaging. So why has mouse been considered and is always propagated to be an inadequate model of human pregnancy? Well, it's thought that it lacks some really important pathology-related behaviors, namely these spiral arteries and their amount and remodeling, especially by trophoblasts and trophoblast invasion. So it's thought that trophoblast invasion is quite shallow, and there's not a large number of spiral arteries in the decidua in the mouse. However, we've learned a lot by this 3D imaging in the heart, and James decided that he was going to do this for the placenta in mice. And so what you're looking at is an image that is going through an entire implantation site where we've stained for trophoblasts in teal and the arteries in purple here. And I hope that you can start to appreciate what we appreciated when we first looked at these amazing images. And that is that there are tons of trophoblasts and tons of arteries quite deep into this tissue, which you can see here. And furthermore, it's really clear that there are a lot of arteries that are completely covered by trophoblasts, just as we see in our human samples. So what James did was to go through and make these 3D analyses of implantation sites from an entire time series every single day until mid-gestation. And then he devised a way in which he could segment them so that he could do quantification of all the different aspects. And what you can see here is that there's a lot of artery growth and trophoblast invasion. And if you look at the specific picture here where we've taken a chunk of this 3D image and highlighted the stain for trophoblasts, you can really see that in this decidua area, there are thousands of trophoblasts. This is really filled with trophoblasts, both in the arteries and interstitia in the stroma. And this is fourfold more than what has been believed in the field from 2D sections. And so typically what people have been doing to analyze the mouse placenta is to take a section that is right in the midline, and then they calculate the trophoblasts that they see in that section. And so that is what James started out doing with his samples, and what he saw was what everybody else in the literature sees. Here you see about 1/5 of the way through the deciduas, the extensive trophoblast invasion. But if you look at these entire implantation sites as a whole, you can see that trophoblast invasion goes all the way up to the myometrium, very, very deep. And also similar like humans, the arteries, many arteries are there at the implantation site, and they also form before trophoblast invasion. And so this is embryonic day 7.5, a few days after implantation. Here's the embryo, and here are these spiral arteries here. They're not invaded yet, but if you look at samples from one more day after this, then you can see these teal trophoblasts are starting to migrate along these spiral arteries here, both close to the artery and also interstitially. And here's just one more view of this 3D view. And so I'll note here that this is something we can't really see with early human tissue samples as of yet. So it's really hard to look in this day-by-day analysis that we can do in these animal models, which is a really big plus to understanding the mechanisms. Okay, so that's the start of invasion. But mid-gestation, a few days later, what we see is that there are many arteries that are fully covered here. This is schematized here, segmented, that is one of these spiral arteries at this implantation site, and the teal is, again, the trophoblast. And you can see here the real pictures through Z slices through these 3D images. And you can see here that they're just completely covered. And if we quantify this, then we see that up to 75% of those spiral arteries that I showed you, those tufts of arteries that are going straight towards the embryo, 75% of these are completely enveloped by this arrangement here. So there is quite extensive invasion of the arteries. So why is this missed in 2D sections? Well, we get some sense of that whenever we do this type of analysis where we take the entire decidua of the implantation site, like so a whole implantation site, and then collapse it and look at it so that we can really get like a circle picture of the entire depth of where cells are localized. And if you look at the density here, where we can see where the arteries lie, will be shown in black here. You can get the sense that there's always arteries, there's always lots of spiral arteries, but they're not always in a regular location. The same is true for trophoblasts. If we quantify the trophoblasts in this way, sometimes they're over here, sometimes they're over here. Each one of these is a different implantation site. And so the co-occurrence, which is where trophoblasts will have invaded and remodeled the arteries, you can imagine here, if you're taking a regular section right through the midline of this whole tissue and just looking at one section, you can really get this idea about how you could miss this, because there's so much variability, because even though you have this relationship between trophoblasts and arteries in every implantation site, sometimes it's over here and sometimes it's over here. So it's kind of randomly biased. So what we are concluding from this is that there is more similarities with humans, especially these pathological important entities between mouse and human. So the spiral oral arteries, there are many, many of these, and then they are highly invaded by trophoblasts, and trophoblasts are invading really, really deeply. And so
Can I ask a quick question? In your sections, you show the enveloping of the trophoblasts, but do you see much replacement of the mouse endothelium with the trophoblasts?
Yeah, that's a good question. So it's not every artery, but there is replacement of some of the endothelium. I think we have yet to quantify that, but I think it's on the list for James to quantify. But we also see that in the human samples that Mira has gotten.
Mm-hmm. And why do you think it's so variable where the arteries are from implantation to implantation site? I would have thought it would have been more consistent.
Yeah, that's a good question. I don't know why. I'll have to think about that more, but that's kind of the way that vessels go. So maybe that's the way trophoblasts go too. It's like vessels, the vessel tree, the artery tree is different from every tree to tree. It's not stereotyped, like the lung, for example, branching. That's because it has this interaction with the mechanical forces of blood flow to tell it how to like shape itself.
Mm-hmm.
There's something like that going on with trophoblast invasion as well. There's some kind of crosstalk that's giving feedback that results in this random bias. Okay, so we're excited now about this finding because we feel like we can really maybe make some important mechanistic findings. And so what we decided to do was to look at a genetic pathway. And because we had done so much work and had so many tools for interrogating CXCL12 and CXCR4, and this has been one of the pathways that I identified as a graduate student in Susan's lab, as well as the atlases that are coming out, are predicting this to be perhaps functioning at the maternal-fetal interface. So again, this is a secreted chemokine, CXCL12 secreted factor by cells that then binds this G protein coupled receptor, CXCR4, and causes intracellular signaling and usually affects migration and those types of morphogenic downstream effects. Okay, so the first thing I want to tell you about is the expression of this ligand receptor pair. And we, over the years, have made lots of tools in the mouse. And so we use these to look at, throughout pregnancy, at the implantation site, during placentation, in maternal and fetal tissues, who is expressing it. And so I'm just going to summarize it here by showing you this cross where we have the fetus, the embryo, in mouse expressing this red fluorescent protein where CXCL12 is expressed. And then the mother, so the maternal tissues, are going to have this mouse that we made where CXCR4 expresses green whenever it is activated, so when it has seen its ligand. And I'm going to show you, this time I'm going to show you tissue section. And so here is early in placentation. This is the embryo. And what we have found is that early in pregnancy, there is the expression by trophoblasts and not the maternal tissues in this area of the ligand, CXCL12. CXCR4 is activated conversely in the maternal spiral arteries. That's not a surprise. This is an artery receptor. And so you can see that here. Here's the arteries. They're forming and they're being activated, potentially by this. And here is the trophoblast marker, but also this is a marker of epithelium. And at this early stage in pregnancy, there's still an epithelium here, a little bit of epithelium. And what we have found is that CXCR4, surprisingly, is also being activated in the epithelium at this implantation site right next to these trophoblasts, as well as some of the stroma around it. So that was a little bit of a surprise. So we have this model in our head now of what might be happening. The embryo is expressing this, and it's signaling to the adjacent epithelium and stroma as well as the arteries. Now the question is it affecting these in any way? And the way that we looked at that is a loss of function experiment. So what happens without this signaling axis? We have many tools for looking at this. The one I'm going to show you first is a deletion mouse, a knockout mouse. It's constitutive, so it's deleted everywhere in the embryo at all times. And so if we have the embryo being knocked out, the mom is heterozygous, and then we can look at this mid-gestation time point. What we see is that, that here, in normal cases, in the sections, we see 20% through the decidua, we can see trophoblasts remodeling the arteries. And then we can see lots of arteries. However, without the ligand in the placenta and embryo, then we see almost no arteries. Here's just one little artery. We see in this section, very few invading trophoblasts. And so we can express that on this type of graph where we're looking at the relative distance of these cell types. This is the myometrium, this is the placenta, and you can see overlap as expected in the controls of trophoblasts and arteries. Again, trophoblasts are teal, arteries are represented in magenta. However, there's this complete separation in the knockout. Okay, so it's important for development of these arteries and seems invasion of trophoblasts. So we wanted to go further and identify the time window in which this was happening. And we could do this because there's a CXCR4 inhibitor. So there's this small molecule AMD 3100 that we can give either fully to look like the constitutive knockout, or we can give early, middle, or late timing to try to pinpoint when this defect is happening. And so as you can see here, the vehicle looks like the controls in terms of where these cells are. The full time point looks like the knockout. And if we look at early and middle, we can see that additively these would look like the knockout. But if we give it late, and this is not really that late actually, it's just our latest, it's E8.5, then this has no effect on placental development here and the maternal-fetal interface. It looks like controls. So this is telling us that spiral arteries, this and other data I don't have time to show, is that spiral arteries develop in this very early window, and they develop through CXCL12, CXCR4 signaling. So when I saw this dramatic phenotype, I was like, how are these embryos living? But if you take out and analyze the embryos from either the drug or the genetic knockout, then you see that they are looking pretty good, especially the 2-day drug, or this is even the full time point drug inhibition. So how are these embryos surviving? Are they surviving without maternal blood flow to the placenta? Well, we turned to 3D imaging to look at this. And what we found when we looked at the midline of our 3D images, we found that, it looks like normally what we kind of intuited from the sections. However, if we would scroll to the other regions, particularly the very periphery, we would see that in the knockout, there was where all the trophoblasts were, and that's where all of the arteries were located, which were interacting with the placenta. And so that's quantified here with this method that I showed you earlier of kind of collapsing this and flipping it so we can see the arteries normally, trophoblasts usually to one side or so, and the co-occurrence. But if we have the knockout, the arteries are way fewer, but still in the middle here. But then there's this peripheral localization of the trophoblasts for some reason, and then very little co-occurrence. However, this peripheral is enough to give you a few arteries going into the placenta. And so if we quantify the number of spiral arteries that appear to be supplying blood to the placenta because they're connected to the placental space, in the knockouts, it is a few arteries. So this must be enough for the embryos to survive. However, what we also saw in the periphery, which was a surprise, is that there were many trophoblasts going really deep, all the way through the myometrium, in fact, and remodeling and interacting with myometrial arteries, so the radial arteries. And that's quantified here. If we take in each implantation site the aggregate of the trophoblasts and look at the percentage in which they're going into different compartments, it's never in the myometrium for the controls, but the knockout has a lot in the myometrium, which is mostly going to the artery. So this is very similar to when I put placental tissue into the mouse fat pad. They just will go whatever's next to it, whether it's what they normally go to or not. Okay, and so this brings about the idea of perhaps, is this a model of placenta accreta? Because this is that situation where I told you there is too deep invasion. So although currently assessing placenta accreta is very difficult, but as this audience well knows, it is usually suspected by ultrasound and then watched. And so some of the things that in the literature are being discussed to look out for are these placenta lacuna, which are a special orientation of these very large spaces within the placenta that people think is because alterations in blood flow are causing these malformed regions. And so that's increasing here in the knockout more than the controls. And also whenever there's a small amount of decidua. So you can imagine that when there's a small amount of decidua, that's when you get major invasion into the deeper layers that it's not supposed to go through. And then if we look at term samples here, we can see then the CXCL12 genetic knockout and in the drug treated, we can see that there is an absence or thinning of the decidua in much of this maternal fetal interface. Okay, so there's also histological analyses that confirm that this is placenta accreta after birth, and that is where trophoblasts are invading through the very depths of the uterine muscle, like almost to the serosa. And we did many experiments to assess whether this was the case, including looking for trophoblasts and actually lineage tracing the trophoblasts. And what we see is in controls, it's never going this deep. This is on the very close to the surface of the uterus. And you can see here that there are lineage traced fetal cells going very, very deep. And you can appreciate how many they are. And they're also sometimes enveloping arteries, just not in this picture. And then finally, the most important part of this pathology to watch out for is maternal hemorrhage and adhesion to the other abdominal organs. And this happens in 40% of the cases in both the knockouts and when we give the inhibitor early. So this is what it normally looks like in mouse pregnancy. There's many embryos here, and this is the intact uterine arteries here, but you can see the hemorrhages here and there is adhesion in this case of the outer wall of the uterus to the other adjacent outer wall, but we also observe it to the other surrounding organs. So this is what James has looked through so far. There's thin myometrium in this model, a thinner absent decidua, these lacuna, very, very deep trophoblasts, adhesions, and hemorrhages. And he's currently doing experiments to assess whether the placenta is retained postpartum. Okay, so what I also want to bring up here is that once we found this, we were very excited that this is maybe a very rare instance in the literature where we have made we have inhibited a pathway just early during placentation, during these very first steps. And then way later at term, then we see these hallmarks of a pregnancy complication. So we are pretty excited about further dissecting and using this model to hopefully learn some mechanistic details about what goes wrong and what trophoblasts are doing in these instances. And one of the first things we wanted to look at was we want to answer the question, is this happening because the spiral arteries are not there? Or is it for an additional reason? And we remember that CXCR4 was not only in the spiral arteries, but also in the stroma and the epithelium. These are important for creating this decidua. And now we're starting to understand that placenta accreta is really perhaps a defect in decidualization. And so we looked at whether the decidua is also forming in our models, both the genetic model and the early injection model of the drug. And so we've inhibited here genetically and then stained these implantation sites. This is E6.5, so this is very early after implantation. And this CDH3, this is a marker of decidualization. And so you can see here, we get the marker induced in normal situations, but in CXCL12, we have a real downregulation of this. And we can confirm this with other markers, including expression of prolactin. And so if we do qPCR on whole implantation sites, we can see both with the genetic knockout and with the inhibition of CXCR4 that we decrease this marker of decidualization. So taking all of this together, this is our working model of what's happening. So the trophoblasts make CXCL12, just like I had seen in my PhD from the human samples. So human and mouse trophoblasts make CXCL12. Then what we think is that this both promotes decidualization as well as signals to and patterns the spiral arteries to be maintained. And perhaps there's also some interaction between this reaction for the spiral arteries. And then this allows for trophoblast invasion to happen, and then setting up the placenta as we know it in healthy pregnancy, which includes, in mice, really, really deep, extensive invasion and remodeling of the spiral arteries. And in the case of CXCR4 inhibition, we are inhibiting this decidualization as well as this spiral artery differentiation. And then so we are seeing defective trophoblast invasion such that they don't go in the normal location, but they peripherally will interact where there's very little decidua and they're going really deep and connecting with the myometrial arteries. And this is causing a number of Creta-like symptoms. So we're pretty jazzed about this, and we're pretty jazzed about going forward to study things like whether this pathway could even be involved in genetic experiments that are done on women with a Creta or not. We're excited that we have this early insult, late phenotype model so that we can do some mechanistic studies in Mira's work on cell-free RNA that you may have heard of. And we're also keen to do screens to where we can identify things that we could reverse this genetic effect. And so that's all I have for you today. Again, this is work carried out by our team, but all the experiments done by James. He's an amazing graduate student. And we thank the Dunlevie for their funding and all the other fundings and everyone else in the lab contributing. And I'll take questions.
All right, Kristy, fabulous. I always love seeing your beautiful imaging and your approach to science. Um, People can just come off mute to ask a question, or if you'd like to put it in the chat, I can read it from there.
I have a question, Virginia.
Yeah, go ahead, Yoel.
Kristy, this was very, very nice talk. Thank you very much. Really exciting. I want to ask you about the phenotype, not the knockout, but the phenotype you're uncovering in the first part of your talk, which is really maybe changing some of our thoughts about the placental invasion, trophoblast invasion, and so forth. In humans, the trophoblast invade beyond the decidua into the inner third of the myometrium. So first question, do you see that? Not in knockout, in the wild type situation. And also, do you see any equivalent of what's called the knitterbook layer in the humans, which is where the placenta separates from the wall of the uterus? It's in the decidua bassalis in the human. It's called after Racia Knitterbook from University of Bern. So I wonder if you see those in a mouse. And also one more question, which is really interesting to me in your knockout. In a mouse placenta, as you know, unlike the human placenta, there's the formation of the canals, which is really a fusion of the spiral arteries within the placenta. And I wonder if those are disrupted in your knockout model.
Yeah, so number one is, we didn't see any invasion into the myometrium. So that is something that maintains as a difference. We only saw that in the knockouts. And number three is what we saw was those canals. Are you talking about? Yeah, I know what canal you're talking about. Mostly those lacuna that were enlarged, I believe, were deeper within the labyrinth zone. But those canals are normally centrally localized. And so the peripheral connections that we saw in the knockout, they're definitely different. But I don't think that James has yet done like a 3D segmentation of those. But that's a really great idea to try to understand how malformed these peripheral connections might be. And you had a second question. What was?
Yeah, it was about a layer of fibrous tissue called the knitterbook layer. And this is exactly where the placenta separates from the wall of the uterus. And that's what assumed to be disrupted in cases of Acrita. Is there a mouse equivalent, or do you know anything about it?
I think we have to look at that, but I have to say that layer causes us problems with our 3D imaging of the human tissue because it gets in the way of our antibodies and stuff. So we have become aware of that, but we did not look for that specifically in the mouse. So I think we should do that. That's a great idea. Yeah, thanks.
Thank you.
Great. Any more questions?
Virginia, there's a hand up.
Go ahead.
Hello. Hi, I have a question.
Go ahead.
Sorry, I think my headphones were going out. Hi, my name is Joy. I'm a postdoc in the Mole Lab, and I'm still getting familiar with this topic as it's pretty new to me. But I have a question around the proposed timeline that you mentioned in like your second to last slide. But do you think that with the differences in timing of decidualization or triggers in decidualization between human and mouse, that like the proposed pathway that you have, like the differences that if you believe there may be differences in the human versus the mouse with that like proposed pathway that you discussed?
Yeah, I mean, we are thinking about that, of course, because we're trying to compare and contrast the mouse to human. And so the thing is the trophoblasts they express this chemokine. The human trophoblasts do make this, but decidualization is happening even before those are around in human. And so I think we've been pretty interested about whether hormones might regulate in humans the expression in some other cell type. So we don't know the answer to that, but definitely interested in figuring it out.
Thank you so much. This is a great talk.
Thanks.
All right. Well, fantastic. Right on time at 1 o'clock. I would like to thank Dr. Redhorse again for a phenomenal talk. and I hope everybody has a fabulous summer filled with fun, great productivity, and some recuperation. And we will see you all back in September.
Great.
Thank you all very much. Thank you, Dr. Redhorse.
Thank you for the great questions.